So we move on to the next speaker. Professor Viola Vogel is from the ETH Zürich. Viola.
I'm very happy that we have the 10th anniversary of CLINAM, and this year so many outstanding discussions also about where we are and where we want to go. And I would like to make one additional contribution to this topic by essentially asking why is it that the EPR effect that was discovered 30 years ago, why is it that it's still the most utilized principle to deliver nanoparticles to tumors? And whether or not, with all the technology that we have available today, whether we couldn't be able to discover new phenomena that could be used either by themselves or in combination to finally not only deliver nanoparticles, but to kill the cells that need to be killed. And we yesterday had already a very interesting session discussing this meta-analysis of nanoparticles and the delivery to tumors. And they analyzed close to 300 research papers, all on mouse, and came to the conclusion that so far we have not increased in major ways the delivery efficiency, and they analyzed a variety of different particles, organic and inorganic, did not see major differences. They analyzed many different tumor models that people have used, and also there have not seen major differences. So I think it's time for all of us to ask, what do we really know? And also to analyze, what do we not know? What kind of new technologies do we perhaps need to analyze and identify and quantify so that we can make better progress? So going back to the meta-analysis, a number of issues were raised that all relate to mechanical aspects. So for example, that the interstitial fluid pressure inside of the tumor is much, much higher than the surrounding tumor, and that pressure gradients and heterogeneous flow in the interstitium are characteristics and strongly correlate with tissue invasiveness and metastasis. Then a lot of the particles are thought to escape the system because they're taken up by macrophages. And coating them prolongs their circulation lifetime, and that increases the probability of the particles to accumulate in tumors. But then we know quite little so far about really all the mechanics by which macrophages pick up particles and how macrophages respond to the physical environment. Then on the next issue that was raised in this paper, even though extravasation occurs, the particles do not penetrate deeply into the solid tumor. So what you see from all the quantitative analyses is, yes, they make it through the vasculature, but then accumulate just beyond and do not reach the cells that need to be killed. So we then saw yesterday, and you see many of these proof of concept studies in the literature, the organ lights up with the imaging techniques that we have available today. But the resolution of these imaging techniques is not that you can resolve whether or not the particles reach their targets. So most commonly, the resolution is a millimeter. And the distance of the particles from the vasculature reaches maybe 10 microns into the solid tumor. So even in the near future, we cannot use current imaging technologies on humans to see whether or not these particles reach their targets. And we need to be aware of the limitations of our imaging technologies. And then in solid tumors, the structural rigidity is supported by extracellular matrix components. And extracellular matrix production, assembly, cross-linking is greatly upregulated. And this is done by fibroblasts and cancer-associated fibroblasts. And also this has not been systematically explored and studied. Furthermore, cancer-associated fibroblasts have many different origins, and you just see from where they can come from. And they have very different characteristics contributing to the heterogeneity in the tumor. And then if you take a biopsy, and since we know how heterogeneous a tumor is from one location, from a particular patient, can you really predict how all the other regions in this heterogeneous tumors will respond to therapy? These are major challenges that we are facing for the years to come. So what are we missing? And I would like to focus a little on tumors and extracellular matrix, because cancer-associated fibroblasts deposit a dense extracellular matrix, and that combined with the pressure can easily close up blood vessels, which makes then these entire regions inaccessible to nanoparticles. What is also known is that in cancerstroma, depending on the rigidity of the environment, one observes that LOX, that's an enzyme that cross-links collagen, and TGF-beta are strongly upregulated. And this correlates with the progressive malignancy of the stroma. So this brings me to next development that happened totally independently from the development of nanoparticles in the biophysics community, and that is the stretching of proteins. And I would just like to show you how fibroblasts stretch proteins. And they reach out, they stretch, they pull, they deform, and as they do so, they deposit new matrix in between the existing fibers. So we should not just view these cells as just sitting there being passive. What happens though, as you stretch proteins, stretching changes their structure, and therefore it can also change their structure-function relationship. All what we know by proteins are equilibrium structures that are determined from NMR or X-ray crystallography. But the way we put together our databases neglects the fact that cells pull on their environment, they can switch the structure of proteins and thereby the functions. And in all the signaling networks that are compiled, very few connections are indicated to be force sensitive. And how this can change an epitope is shown in this steered molecular dynamics simulation, where a peptide binds to fibronectin, and you see how the stretching of this peptide, of the fibronectin fiber, can destroy the epitope, by which in this case S. aureus binds to fibronectin fibers. Just to illustrate that by stretching proteins, you can switch into a different function and then put perhaps signaling pathways. So the field of stretching proteins started with the atomic force microscope and optical tweezers, so it's at the core of nanotechnology. We have learned a lot by using these nanotech tools about how proteins are stretched and that there are many structural intermediate states that might have different functionalities. We learned a lot in cell biology about mechanobiology, that intra and extracellular proteins are working as mechanochemical switches. But then this does not so far connect at all to the tissue level scale, because we do not have tools to study that at an organ level. So we do not have tools to measure forces in organs. So how much force does a cell apply to its environment? How much does a cell stretch the fibers in its environment? And therefore we can't study these phenomena at the organ level, despite the fact that so much progress has also been made in that field at the molecular and the cellular level. So why is it so important to go into this question? This is just one protein, just one extracellular matrix protein. It's a multi-domain protein. It's fibronectin. It's highly upregulated in wound healing, in embryogenesis, and in cancer. You see it has many binding sites for all the growth factors that we constantly deal with when dealing with cancer, for MMPs, for other extracellular matrix proteins, and so on. So stretching could potentially destroy each of these epitopes or maybe open up new epitopes that we are so far not targeting with our equilibrium-based screening technologies. So are proteins stretched and unfolded by mechanical forces generated by cells? Today we know yes. 15 years ago, it was still highly, highly debated. So in order to show in cell culture that stretching can indeed partially unfold proteins, we developed a FRET probe a couple of years ago and then added this to the cell medium. The cells incorporated, harvested out of the medium, incorporated into the extracellular matrix. And here you can see that extracellular matrix produced by fibroblasts is very heterogeneous in terms of its strain, the extent to which it's stretched by cell-generated forces. So the blue color indicates that it's stretched 3, 4, 5 times the equilibrium length from relaxed fibronectin fibers. So does it have an impact on cell signaling? We analyzed that so far on the, with respect to the osteogenic differentiation of human mesenchymal stem cells. And we generated fibronectin fibers, single fibers, seeded stem cells onto these fibers, and then cultured them under different differentiation media, either on relaxed fibers or on stretched fibers. And what we see is that osteogenic differentiation is considerably upregulated on stretched fibers with respect to relaxed fibers. So the fibers are stretched by cell-generated forces. So it's this reciprocity between the cells pulling and then responding to the fibers. So we are thinking that stem cells might use the strain of fibers as a checkpoint to determine their future pathway. And what we found was that they recognize stretched fibers mostly through the EGFR signaling pathway. Next question. We all know that collagen matrix is deposited excessively in cancer, but also in fibrosis. But how do the cells control where these fibers are deposited, in what locations? So for that, it's interesting to note here that, I wonder whether I can show that, that fibronectin has a collagen binding site, and it's circled on the slide. And we asked whether or not the binding of collagen is regulated by the mechanical stretching of extracellular matrix. So again, we did this in cell culture. We added FRET-labeled fibronectin to the medium. It's incorporated in the matrix. You see some regions highly stretched, and some are more stretched than others. Then we labeled collagen antibodies with a third color. And what you see is that the collagen in early stages of the matrix co-localizes with fibronectin, but it only co-localizes with the relaxed state of fibronectin, not the stretched state. So just remember that for the next slides to come. But then as the extracellular matrix matures, we see that the cells start building up more and more collagen, and finally they start pulling on collagen. And by FRET, we see how that relaxes the fibronectin matrix back to a relaxed state. And then if you would digest the collagen matrix, we see that the cells pull again on fibronectin, and the FRET is again reduced. So how can we utilize all that information? Big challenge ahead also of this field is that we need to bridge the length scales effectively to make a contribution. And ideally, we would like to bring these two fields together. So the question we asked ourselves is, can we generate peptide probes that can measure the strain in tissue of fibronectin fibers? One common approach might have been to take high-throughput screening approaches, but we want to take advantage of nature's evolutionary process. And here are all the bacterial peptides that are known to bind to the N-terminus of fibronectin, which is shown here in the video, and I hope it plays. So it's a multi-domain motif to which all these bacterial peptides bind. They bind in antiparallel beta sheets. And if you start to stretch this segment, you see how the distance between these domains increases. And this is the mechanism by which bacterial peptide affinity to fibronectin is reduced upon stretching of the fibers. We then synthesized peptides from bacteria and synthetic ones after understanding the motif. All this started with a steered molecular dynamics simulation that you've seen. And we could then indeed see on the stretch assays that the peptide binds to the relaxed but not to the stretched fibers, the relaxed fibers being greenish red. So we next asked, and can we validate now in cell culture that this peptide is working? So we combined our FRET probe and the peptide and added then peptides to the cell culture medium. And again, we see that the high peptide concentrations in pink correspond to the regions that are red in cell culture, so they co-localize. So the next question we now ask are fibronectin fibers under higher or lower tension cancerstroma. This question has never been addressed because there was no tool. We therefore used the peptide, this bacterial peptide. We used an antibody that colors the entire cross-section, histological cross-section through this cancer tissue. It's rich in fibronectin, but only some regions are really rich in relaxed fibronectin fibers. So far, the physiological significance is not at all known because we never could even look into these mechanoregulated processes at the tissue level. And finally, I would like to say that we can also inject these peptides into mice, and they are stable for several days, and they accumulate again in cancer stroma. And with that, I would like to say that we develop 3D cell culture models now to microtissue platforms to test all these dependencies. And with that, I would like to close saying we need new physical tools. We need to combine what we have already to make progress in the field, to quantify and understand new phenomena, and then combine this to have a bright future in the field of nanomedicine. With that, thank you to my research group and the many collaborators, and thank you to you for your attention.
Thank you, Viola, for this inspiring talk and very timely as we try to understand cancer biology and may have to refine our understanding of EPR in the end. Questions?
So very interesting. I'm an immunologist, so I'm interested in the power of these different fibers to stimulate immune responses. As you know, the collagen 1 is made into sort of large fibrils wrapped around a core of collagen type 5. And we're very interested in collagen type 5 because it's unlike collagen 1, which we're very tolerant to. We are very reactive to collagen type 5. It's extremely immunoreactive. So the question was, as you stretch the collagen 1 fiber, is the exposed inner core now available to the immune system?
You're raising a really important question, which I can't answer. I can only say that we look in the moment whether or not fibronectin fibers are more or less stretched if you allow a lymph node to swell. And we also asked the question in the moment whether IL-7, which binds to fibronectin, whether that binding is regulated by the mechanical tension of fibronectin, and we see indications that this is the case. So one question, there's so incredibly much that needs to be learned on how these phenomena are regulated by forces and pressures.
One last question there. Okay, then we...
This is a curiosity. I don't know if it's very much related, but there are people that they have ulcers, particularly in diabetes, and everybody is looking at the role of the fibroblast. And it seems that the metabolism is completely changed. But could it be that maybe many of us, we are looking to the wrong place, and that this may be the matrix that is generated by this fibroblast that is completely different and is basically not allowing to get this reconstitution? I said this because I saw something of wound healing in your slides. I don't know if you could comment.
So this is a question again because of the fast increase of diabetes that we are planning to address. Particularly how fibroblasts, we needed to get a license now that we can take biopsies from patients and get those fibroblasts for a comparison. These studies are coming.
Thank you very much again